Hi, my name is Ravali Adusumilli. I'm a research analyst in the Malik Lab at Stanford University. Today I'm going to talk about challenges in reproducibility of multi-omic analysis. So, what are the current challenges in multi-omic analysis? First, there's a lot of data. Second, the data is continuously growing. Third, the domain knowledge is isolated. And finally, data analysis is complicated. A simple analysis has many steps, and each step can be performed by multiple tools with dozens of parameters. To investigate the challenges in reproducibility, we attempted to reproduce Zhang et al.'s multi-omic study of colorectal cancer. This is a proteogenomic study across proteomics, transcriptomics, and genomics with data from TCGA and CPTAG. When we started the analysis, our initial questions were, can we reproduce the results from the paper? And how easy or hard is it to reproduce the results? Then, can we assess the sensitivity of the pipeline to the results? And like discussed earlier, the data is continuously growing. So how does the new data impact the results? We used a semantic workflow system called WINGS for our analysis. WINGS was developed at the Information Sciences Institute at USC by the Gill Lab. Using WINGS, we used multiple tools and scripts to reproduce the results by developing workflows. Each workflow is made up of several components. So if you look at the left, this is a sample workflow. Anything in gray is a component and anything in blue are the input or the output files. So let's consider a sample component. In the above component, we can add species name as metadata for the files. And then at the component level, we can set up rules using Jenner rules. This way, when the workflow is executed, only the data compliant with the rules is selected for execution. If no such data is selected, the workflow cannot be executed. Here are a couple of workflows from WINGS which we used for our analysis, the proteomics and the proteogenomics workflows. While trying to reproduce the results from the paper, we've had to repeat our analysis a few times. So our data analysis was performed in 3 different stages. In stage 1, we followed the data processing instructions from the paper only. In stage 2, we obtained some instructions from the authors and also had to change one of the reference files that we had used. In stage 3, we used the same reference protein sequences database files that the authors provided and continuously communicated back and forth between the authors, received a lot of scripts, and verified the parameters. So as you can see, our very first attempt at reanalyzing the data produced dramatically different results from the original analysis. We identified 11,500 protein groups as opposed to 7,000 protein groups in the original analysis. And at the mRNA and protein abundance correlation level, when we compare between the 2 data sets, the correlation was about 0.884. For stage 2, the number of protein groups increased quite a bit, but we still had a difference of 1,500 proteins. At the mRNA protein abundance correlation level, actually the correlation between the datasets fell down a little bit. So in stage 3, again the protein groups increased a little bit. We still had a difference of 1,000 protein groups, but the mRNA protein abundance correlations actually increased quite a little bit compared from stage 1 to stage 3. So stage 3 was as close to the original analysis as we got. After looking at the protein groups, we also wanted to look at the peptide groups. Because at the protein groups, the difference was 1,000 protein groups, but at the peptide groups, it was 23,000 distinct peptide differences. So we also looked at the single amino acid variants next between the customized and standard database approaches. And the differences was about 600 SAAVs between the data sets. So after looking at the mRNA protein correlation again, we wanted to look at the final results of the paper. And we found that the subtypes between both the original and our analysis were very similar, except a couple of samples. One was present in the original and absent in the reanalysis, and one was added in the reanalysis and absent in the original. But by large, they remained very similar to each other. So next, to assess the sensitivity of parametric variations on the results, we compared the proteogenomic and proteogenomic workflows using standard and customized databases. When we look at the proteins identified between the reference and the custom data sets, the difference was about 2,307 protein groups. And then we use spectral counts to calculate the correlations for the overlapping proteins in the protein group. protein groups amongst the data sets. This resulted in an average Spearman correlation of 0.86. Then, to assess the sensitivity of a search engine, peptide search engine, we performed 2 independent searches using mirror match search engine. We altered only one parameter between the 2 searches. This resulted in a difference of about 627 protein groups and 3000 peptide groups. And last, we used 4 different search engines to identify proteins independently. Each search engine identified different number of proteins. However, most number of proteins were identified by all 4 search engines, suggesting that a complementary approach is probably the most effective. Over time, new colon cancer data was made available on CPTAC. So we processed 20 new colon cancer samples in 5 sample increments to the data set. The 20 new colon cancer samples are highlighted in brown. Post-batch correction, the new and old samples merged into the subtypes together, but few of the original samples were absent in the newer by adding the newer samples. So the samples shuffle around a little bit in the subtypes. And to conclude, based on our analysis so far, most of the differences we observed can be attributed to algorithm randomness, such as in the clustering case, and software parameters that are hidden, and differences in reference files, unavailability of the reference files of the same version, and the unavailability of software, and lack of proper documentation. And despite the changes in the lower level, the highest level findings are still very well reproduced. Also, using the semantic workflow system, we could radically simplify the analysis, explore similarities and variations quite easily, and also process the new data and continuously update the final results. So everyone who helped us, thank you.